Thank you to everybody for being here, and special thanks to our local partner here in Israel, Limed. First of all, my presentation will be focused on the biological evaluation of medical devices and the main changes and updates of this standard that will represent the challenges for the manufacturer of medical devices. Before starting with the technical part of the presentation, I will give you a couple of information about my company, Eurofin. Eurofin is a worldwide network of laboratories, more than 410 laboratories worldwide in Europe, US, Asia Pacific, and more than 42 countries. It was founded in 1987 and then now has 30,000 employees all over the world and 3 billion revenues and we are a world leader in food, environmental and pharmaceutical product testing, and with more than 20 years of experience in the medical device world, we created a division called Eurofin Medical Device Testing that is composed by 16 laboratories worldwide and 3 centers of excellence, one located in Lancaster, Pennsylvania, one in Munich, Germany, and the other in Milan. So basically, thanks to that work of laboratories, we are able to support our clients during the whole life cycle of a medical device with a complete portfolio of tests: microbial testing, toxicological testing, chemical and testing, and also physical and mechanical one. But let's start to talk about the main changes of ISO 10993 with some information at the beginning regarding the medical device regulation. So according to the medical devices regulation that was issued last year and will come into force in 2012 after 3 years of a transition period, manufacturers should demonstrate that their medical devices are safe and are effective. So they must satisfy general safety and performance requirements included in the European regulation. In fact, despite all the beneficial effect that a medical device can have, for sure, the contact of the medical device with the body can generate some risk that should be evaluated. And even if materials that compose the medical device can be really well-known materials in other fields like food-grade materials, their combination in a medical device, the manufacturing process, the sterilization process, and also all the other post-manufacturing process can in some way modify the characteristics, the features of these materials and generate some concern in terms of biocompatibility. That's why the manufacturer should demonstrate that their devices are safe. But how they can demonstrate how can they satisfy the safety requirements according to the regulation? Basically, there is no mandatory pathway to be followed. I mean, from theoretical point of view, a manufacturer can do everything he wants to demonstrate the safety of his device, but for sure, he needs to apply an approach that is time-saving, cost-saving, and that should give a robust proof, a scientific proof of safety, and should lead a really smooth approval process with the competent authorities to obtain the CE mark, for example, or to succeed FDA submission. That's why ISO 10993-1 standard is really important, because according to the regulation is considered the state of art in terms of biocompatibility assessment. So basically there is no real good reason not to use this standard to demonstrate the safety of a device. Because basically, if a medical device is in compliance with the ISO 10993 series, manufacturer obtain the presumption of conformity to the general safety requirements of the European regulation. And so this standard is really important in Europe, but it's also really important for the US because it's also in this case considered the state of art for biocompatibility assessment. But if you are going to US market, you need also to consider that the ISO 10993 is not sufficient because you should also consider the guidance that FDA issued 2 years ago about the use of ISO 10993-1, because FDA has his own interpretation of the standard and provided also additional requirements for the biocompatibility. That's why you should consider both of them. But what is the ISO 10993-1? It's an horizontal standard, so it means that can be applied to all medical devices, to the whole categories of medical devices. It was issued in 2009 and basically describes the general principle that governs the biological evaluation, that is considered a part of a structured program within a risk management process according to ISO 14971. And thanks to this part of the ISO 10993, we can identify the categorization of medical devices according to the nature of contact and the time of contact. And thanks to this categorization, we are able to identify the biological effect that there can be considered relevant for our medical device. And for sure, considering all this information, physical and chemical information of medical device, already existing data about the toxicology, about preclinical tests, and performing the tests which protocol are included in the ISO 10993 series standard, we can demonstrate that the device is biocompatible. Now, this standard is under review, and we are really lucky because we have our colleagues involved in the ISO TC 194. It is the group that deals with the changes of ISO 10993. So we have some anticipation for you regarding the new requirements of ISO 10993-1. Now we have a committee draft. This committee draft will become a final draft in the next few weeks, and probably in 2-3 months we will have the new standard. So let's start to talk about the main differences between the current standard, so the ISO 10993-1 of 2009 standard, and the committee draft. Let's start to talk about the medical device categorization. Basically, according to the ISO 10993, we have two kinds of categorization. Categorization of medical devices according to the nature of contact and according to the duration of the contact. According to the nature of contact, we can have 3 subcategories of medical devices. We have the surface contacting devices that are in contact with the skin, for example, or with mucosal membrane or the breach or compromised surfaces. We have also the externally communicating device that basically are placed totally or partially outside of the body, but have a contact with the inner part of the human body. And then we have the implant device. So, according to the categorization by nature of contact, now the committee draft includes a new category called non-contacting device. In this category, there are all the devices that, for which the biocompatibility assessment is not necessary, like, for example, a diagnostic software, because there is no direct or indirect contact with the human body, so the biological evaluation shouldn't be demonstrated. And according to the categorization by the duration of contact, we have 3 categories of medical devices. Limited exposure medical devices, when the exposure lasts less than 24 hours. Prolonged exposure medical devices, when the contact is between 24 hours and 30 days. And then we have long-term devices, when the exposure is longer than 30 days. In this case, the classification is basically the same. Some changing in the words. In the past it was called permanent contact when the contact was longer than 30 days. Now we are calling long-term contact medical devices, but basically it's the same. But now the committee draft included another category of medical devices after providing us information about the categorization by the nature of contact and the time of contact. It is composed of the transitory contacting device. So all devices that have a really brief and transient contact with the human body, and for which basically biocompatibility is not necessary, except for some cases in which even if the medical device is a really brief contact with the human body, some compounds, a component of this device can remain in contact with the body, like for example coatings or lubricants. In this case, even if the contact of the whole medical device is really short, we need to evaluate the biocompatibility. But now we will focus on the most important change of the ISO 10993-1. In the present picture, you can see the screenshot of the table included in the current standard. This is a really, it's a really useful table because based on the categorization of the medical device according to the nature of contact and duration of contact, you can identify the biological effect considered relevant for them. But up to now, this table was considered a sort of checklist. I mean, I can categorize my medical device in this way. Okay, I need to perform this test, this test, this test. If I perform all these tests, I'm okay. So now the approach is changing a lot. This checklist approach is no more suitable because it doesn't consider a lot of important issues related to the medical devices. And for sure, it doesn't allow a full understanding of our device. So basically, these biological effects are considered a sort of a suggestion, and the table should be considered as a framework through which we are able to identify the biological effect considered relevant and that we should assess. But it's not necessary to perform all the tests for all the endpoints, because in this case, we can lead to unnecessary testing. So this is the reason why now the table is changing a lot. Also, at the first glance, you can see that it's really different from the previous one because there were included a lot of columns and a lot of information about the endpoints of the biological evaluation. And in the next slide, we will focus on these differences, but let's start to think about the title of the table. In the past, this table was called Evaluation Test for Consideration, so it was really focused on tests. Now this table is called Endpoints to be addressed in a biological risk assessment. So as a first glance, we can we can understand how the biological risk assessment is now really, really important and play an important role in the biological evaluation and demonstration of the safety of our device. If we zoom the table in some parts, we can see that, for example, the first column that is now included in the table is related to the physical and chemical information. So, physical and chemical information about the medical devices are now the first step of the biological evaluation. And in particular, they are marked, they are requested for all kinds of medical devices, as you can see in the table. Basically, even if you have a skin contacting device, an implant device with permanent contact, you need to provide this information. And moreover, these information are considered a prerequisite for the risk assessment. That means that you should provide this information, you should collect this information before performing any kind of biological testing. So it's really now important. And how we can address this endpoint? For sure, we can consider the ISO 10993 series, and in particular ISO 10993-18 series, 18 for the chemical characterization, and ISO TS 10993-19 for the physical to obtain physical information. So basically, the idea now is that before performing any kind of test, especially in vivo tests, we need to improve our knowledge about our medical devices in terms of composite materials, manufacturing process, sterilization process, and all the other processes that can influence the biocompatibility of our device. And how we can perform a chemical characterization? Basically, the extent of a chemical characterization depends on the nature of contact with the human body and the duration of contact of human body. And depends also for sure on the physical nature of our device, because we cannot think to characterize in the same manner a gel, a powder, or a solid. So all this information should be combined to identify the right approach. Basically, there are two kinds of chemical characterization. A direct material characterization that is focused on the inner chemical properties of our medical device, and then the extract characterization that is focused on the substances that can be leached out from our device, not only to the composite materials, but also to the manufacturing process agents, to the sterilization process, and so on. And there are a lot of analytical techniques that can be used to achieve this goal. But for sure, the most extensive chemical characterization can be performed through an extractable and leachable study that through which applying different analytical techniques like GC-MS, LC-MS, ICP, we are able to identify the semi-volatile, volatile, non-volatile organic compounds, and also all the inorganic compounds that can be released from our device. And only after collecting all this information we can think to the other endpoints to be evaluated. So, for example, cytotoxicity, sensitization, genotoxicity, and so on. And basically, thanks to the chemical characterization that now plays an important role in the biological evaluation, some tests can be waived. Because, for example, if we perform an extractable study, we identify all the compounds that are released from our device, organic compound, inorganic compound, and we evaluate them from toxicological point of view, and we can conclude that the amount released from our device is safe. We could not perform some biocompatibility tests, especially the tests related to the systemic effects, so subchronic toxicity test, chronic toxicity test, genotoxicity, carcinogenicity, that we know that are the most expensive and the longest tests. So it's a really new and good way to assess this endpoint through the chemical characterization without performing the test, in vivo test. So the approach is changing a lot. So we are moving from a checklist approach in which we need to perform all the tests to a really more structured program of biological evaluation in which, first of all, we need to collect all useful data regarding physical and the chemical information. And then evaluating from toxicological point of view this data, we can decide if further tests are necessary or not. And just for your information, now the ISO 10993-18 that deals with the chemical characterization and ISO 10993-17 that deals with the toxicological evaluation are now under review. Another column that was included in the ISO standard was related to the material-mediated pyrogenicity. Pyrogenicity is the ability of a chemical to induce a febrile reaction and can have different sources like materials, like endotoxin, like also gram-positive fungi. And by now there is no validated test for medical devices that is able to cover all the sources of pyrogenicity. But for biological evaluation, material-mediated pyrogenicity is requested. And the way to achieve this endpoint is to perform the rapid pyrogen test. So now, also ISO included this column that was already included in the matrix that the FDA issued 2 years ago in the guidance. So now ISO is totally aligned with the FDA requirements in terms of pyrogenicity. And then, also the long-term effects were clearly included in the standard because in the past there were mentioned only subchronic, subacute toxicity test and genotoxicity, and the other were mentioned in some part of the standard, but now they are clearly included and identified in this table. So also they are really, really important now. And last but not least, the ISO standard provided additional information on nanomaterials, absorbable materials, and briefing gas pathway medical devices, providing the appropriate reference to specific ISO standard. So in the next few months, we will have a new ISO 10993-1 standard. And as you can see, it changed a lot if compared to the previous one. So my suggestion is to be prepared because it's a real challenge for the manufacturer. And if you are prepared, you can save time, you can save money, and proceed with a really smooth approval with the competent authorities. So my company, Eurofins Medical Device Testing Division, can support you in this kind of evaluation because we are a leader in the testing of microbiology, biocompatibility, toxicology, chemical characterization, and extractable leachable study. And we can also provide you regulatory support in terms of biological risk assessment, so biological evaluation plan, biological evaluation report. So for any further question or need to assistance, feel free to contact me or Sally, that is our local reference here in Israel. So thank you very much for your time, and I hope that the presentation will be useful for us. Thank you.